Hello, welcome to my coronavirus classroom. I'm Janessa Jacobs, and this is Nervous Tissue Part 2. So today for our bone of the day, we're going to look at the strongest bone in the body, the femur. This is the long bone of your thigh, not your leg. Your leg has two long bones in it, just like your forearm had two long bones in it. So the femur is what we're talking about today. And yeah, so this is the thigh, or the femoral region. As far as how are we going to know if we're looking at a left or a right femur, this happens to be a left femur. The head of the femur is going to plug into this really sturdy socket right here called the acetabulum. And so that has to go medially, as you can see here. Kelly is showing us that the head of the femur must go medially. So you could say, okay, yeah, but does it go medially this way or does it go medially this way? And the answer is this one goes medially this way, and I can tell because if the head of the femur is superior and I look at the inferior or the distal part of the femur, I can see this nice smooth articulating surface right here. The patella, or your knee bone, is going to sit right here, and so we need a nice smooth articulating surface for that. If you look at the posterior aspect of the distal end of the femur, you can see that there's this huge notch right here, and that's not going to articulate nicely with a knee. So this means this is anterior, this means this is my head of my femur facing medially, so this is my left femur. I'm too short to show you that on my leg. In class it would make a lot more sense. So as far as the markings that you need to know on the femur, this is the head, this is the neck. We've got a greater and a lesser trochanter, and the femur is the only bone with trochanters. And so the greater and the lesser trochanter, then we've got our shaft, and then the medial condyle and lateral condyle. So condyles are smooth articulating surfaces, so we can figure out which is medial because the medial condyle is going to be distal to the head of the humerus, which is pointing medially, and the lateral condyle will be then lateral to that. So those are all of the markings that you need to know. That is your femur, and you do need to know left versus right. Yes, on Kelly or on a picture of this femur or from something from Mastering A&P. Any of that is fair game for your practical. This practical is a beast. You have 30 bone stations and, 30 questions, 15 bone stations, and 15 muscle stations. That's how it works out in class anyway. And it's 30 points each, so 60 points for that practical. It's a lot. That's why we're doing bones and muscles of the day now, so that you're not completely overwhelmed at the end. All right, for our muscle of the day, one of the things I want to say about the muscle of the day is my poor students in face-to-face classes, if they're not closing their eyes during muscle meditation, I stare them down. Because I'm not closing my eyes during muscle meditation in the face-to-face class, because I don't have to. I know my muscles. I meditate all the time. That's, I don't know, kind of a lie. But I muscle meditate. And so yeah, I might just start staring at you during muscle meditations. Your eyes closed? You're supposed to be doing this stuff. Like, come on, people, pretend we're in class. You're getting the face-to-face value online. That's only as true as it can be. Okay, so we're actually going to meet 4 muscles today, and that's because they share an action. One of them does a little something else, and these are all the muscles that we find on the anterior aspect of our femoral region. We just met the femur that's deep in here, so we might as well talk about the muscles that are sitting on top of it. So right here, these 4 muscles right here in the front are grouped together and called the quadriceps femoris. Femoris because we find them in the femoral region, and quadriceps because there are 4 of them. We can only see 3 of them right here because one of them is deep. So let's just quickly talk about the names of them. I'll pull it off and then show you the deep one, and we'll talk about what they're all doing. So rectus femoris is this one right here. And all of them are leg extensors, which means that if your leg, which is down here, is flexed, and you want to extend it so that right now this leg is extended. So the muscles that contracted to extend your leg were the quadriceps, and they contract and pull on your leg and you extend it. So rectus femoris extends your leg, but it also helps with some flexion of the thigh. So flexion decreases an angle. So if you were in anatomic position right now and you were to flex your thigh, rectus femoris helps with that. There are other muscles that are thigh flexors that we'll talk about. I don't think we do them as muscles of the day. I think we'll do them when we get to the muscular system. So anyway, rectus femoris helps to flex the thigh, which is this, and helps to extend the leg, which is this. Okay, the other three quadriceps are vastus medialis, vastus lateralis, and then deep to rectus femoris, we have vastus intermedius. So these are our four quadriceps, and all of them are leg extensors. So you only really need to know one action for 3 of the 4 quadriceps, and then just know one more action for rectus femoris. So again, rectus femoris extends leg and flexes thigh, vastus lateralis extends leg, vastus medialis extends leg, and deep to that is vastus intermedius extends leg. We're going to breathe through each of them, their name and their action twice, and then call it good. All right, so close your eyes and breathe in rectus femoris, and breathe out flexes thigh, extends leg, and breathe in rectus femoris, and breathe out flexes thigh, extends leg. Breathe in vastus medialis, and breathe out extends leg. And breathe in vastus medialis, and breathe out extends leg. And breathe in vastus lateralis, and breathe out extends leg. And breathe in vastus lateralis, and breathe out extends leg. And breathe in vastus intermedius, and breathe out extends leg. And breathe in vastus intermedius, and breathe out extends leg. And breathe in quadriceps femoris, and breathe out extend leg. And breathe in and breathe out, and come back to class. All right, so I'm going to start out with a couple review questions for you, and then I want to review a couple other things that we touched on last class so that we can move forward and all be on the same page. Okay, so first, neurons can be functionally classified as A, sensory, B, motor, C, interneurons, D, all of the above. All right, the answer is all of the above. They can structurally be classified as, I might have that question later, so I'll be quiet for now. Okay. The door shuts loudly behind you and you flinch. Your heart rate increases as a result. This response is due to the activation of your nervous system, of your blank nervous system. A, autonomic, B, somatic, C, central, D, all of the above. All right, the best answer for this question is autonomic. And let's talk about why. Let's work our way through these answers, because you could say, well, heck, lady, you only talked about the autonomic nervous system for just like a brief moment yesterday. And that's true. But still, we did say that autonomic, we talked about like sensory information, somatic being what you're aware of, visceral what you're not aware of. And then we talked about the efferent division of the nervous system, and in the peripheral nervous system, how the somatic division was what was innervated by or innervating our skeletal muscles, and that's the only thing we have control over. I then said we have an autonomic division we're going to talk a lot about at the end of the semester because it controls everything you do next semester. You can put that information with the other information that we said is that neurons, the effectors, for those motor neurons, the effectors were either muscle or glands. The type of muscle is either skeletal muscle, which we have control over, or cardiac muscle, or your smooth muscle. And so if your heart rate is increasing, what we're doing is increasing the firing of cardiac muscle, and you are expected to be able to put all of those pieces together. And that may seem like a lot to ask you, but you're all going into healthcare, and your patients are going to walk in and they're going to be like, oh, my belly button hurts. And they're going to be whining and they're going to want drugs, and you're just going to be like, there's nothing wrong with your belly button, it's your appendix. And you're gonna know that because you're gonna have to like put all this stuff together. So yeah, just be aware of that. Okay, I wanted to just review a couple things to make sure that everybody's comfortable. So when we move into the action potential, there aren't kind of any questions about what's going on. So we said yesterday that our neurons are going to be able to use this resting membrane potential that they have to do something, because unlike most other cells, our neurons have voltage-gated channels. And so we talked about some definitions of voltage and current and all of that. I don't want to go over the definitions again. I just need to draw your attention to some basic overarching like laws that guide like positive and negative charges. So the first is that it takes energy to separate opposite charges. Well, I guess that's the second. The first is that opposites attract. So positive charges want to be next to opposite charges. And the second principle is that then if you have to keep them apart, it's going to take energy to do that. So first, opposite charges are attracted to each other. And because of that, if you want to separate those charges, it's going to take some energy. So separating those charges requires energy. And so why are we talking about this? Well, because if we look at the plasma membrane, it's a phospholipid bilayer that is going to separate charges in our body. If we look at the inside, it's relatively negative compared to the outside, which feels relatively positive. And the reason for that is we've got negatively charged proteins that get stuck inside the cell and they can't leave. They hang out here near the plasma membrane, and so sodium on the outside is attracted to this. Potassium is on the inside is also attracted to this negative charge because positive charges want to be next to opposite charges. So right here at the plasma membrane, what we have is a system where we've created potential energy. Why? Because separating these charges requires energy. So the next basic principle of electricity that I need to address is the fact that then if we have separated these charges, when we move them together, energy is liberated in the system. So this separation of charge gives us potential energy. That's why it's a resting membrane potential. We can potentially do something with it. So the last thing that we'll say is that energy is released when opposite charges move toward each other. So this system in the neuron, well, all of our cells, because all of our cells have resting membrane potentials. And we talked about that yesterday. You can go back and review that, or go back and review chapter 3, section 5, and go all over that. But neurons are going to have these channels that are going to open up to allow the movement of ions through here, which is going to conduct a current. Yesterday we said current is the flow of ions. That's a charge. We're moving these charges and it's flowing down this neuron so that we can conduct an electrical potential. That is so cool. I love it. And I just wanted to make sure that we're all on the same page about these basic principles of electricity and how they're going to guide the movement of these two ions in an action potential. So, just some quick things to kind of go over and put in our back pocket. Sodium is higher in the extracellular fluid. Potassium is higher in the intracellular fluid. Okay. Okay, and what keeps this different concentration is the sodium-potassium pump. Okay, if we stick a voltmeter in here and measure this negative difference, what does it feel like? In a neuron, it feels like negative 70 millivolts right here at the surface of the membrane. Okay, so if I move away from this and I have some kind of channel that can open in response to some voltage other than negative 70 millivolts, then I'm going to be able to start a current or a flow of ions so that I can get this electrical signal going in a cell. Okay, that's where we're going. That's what we're doing today, and it can be one of the more complex things to wrap your head around, but then once you get it, it totally makes sense, and it's so cool. This is one of the coolest things that happens in the body, and what's great about it is that all you have to do this is stick proteins up in the cell membrane. So when we were talking about cells that first day, and I said, you know, like channels, these are these proteins that you stick up in a membrane, and what a channel does is facilitate the diffusion of an ion down its concentration gradient. We're going to be talking about channels right now. They're just proteins. They don't have any choice except to behave the way that they behave, and we'll talk about that in just a moment. And when they open up, when some of them open up, sodium's going to rush in, and we're going to get a positive flow in here, and it's going to go really positive. And then other ones are going to open up and take over, and potassium's going to flow out, and positive charge leaving is going to make us get negative again. So we'll have this wave of positive followed by negative that's going to happen because of the behavior of these proteins, who have no choice but to do what they do. So if you can start wrapping your head around it, what each of the proteins is doing, and then how the ions are moving through it, you should be able to put together a cohesive picture of an action potential or a nerve impulse. Okay, the other thing that I kind of wanted to review was the operation of these channels. So I'm not going to go over the definitions again, but you've got a figure in your book. I'm not copyright infringing is why I'm not using the figures from your book. I don't believe in that stuff. I'm a writer, and I don't want people to copyright infringe from me. So yeah, look at that figure in your book. The point is, with a gated channel, when it's got two states, or it can have three states in the case of the voltage-gated sodium channel. I'll talk about that in a little bit. But when it's closed, so it's just closed and nothing is flowing through. And so you could say that right now our gated channel is closed and nothing is flowing through this channel. And then our chemically gated channels are going to be able to open when a chemical binds. So the chemicals that we're going to be talking about right now are neurotransmitters. So what happens to this type of channel then, or in this type of channel, is a neurotransmitter will bind its receptor. So there will be some receptor here for the neurotransmitter on this channel. So this is a ligand gated or chemically gated channel. It's also a neurotransmitter receptor. So when the neurotransmitter binds to its receptor, it will open this gate. That's the key to the gate, is neurotransmitter. Chemically gated channels. The key to their gate is a chemical that's a neurotransmitter. When it binds, this gate opens. So I can't like move this gate to open it. I can't shock this gate with a voltage to open it. Those aren't its keys. I can put a very specific neurotransmitter on it. And I can't just put any old willy-nilly neurotransmitter on it. It has to be the neurotransmitter that's specific to this receptor. But once I do that, the gate opens and an ion is going to move down its concentration gradient. So now this gate is open and an ion flows. So ions have charges. A flow of charges is a current. So now through this gate, I can cause a current. These gates are going to cause those graded potentials that we talked about last time, and we'll talk more about them later in the summing of graded potentials. So chemically gated channels cause these little graded potentials that are going to just be these little short-lived, either depolarizing or hyperpolarizing events. Okay, ions, that brings up another good point, which is if you haven't had a chemistry class in a long time and you didn't, or even if you haven't had a bio class in a long time and didn't review it, maybe you don't remember what ions are. Ions are charged particles. I need a periodic table of the elements right there. It's decided right now. So what happens is that you can get these like sodium, Na+. Why do I write it this way? Well, because it's given up one of its electrons and it feels a little positive. So an ion that feels a little positive is a cation. Cations have positive charges. So sodium's a cation, potassium is a cation, calcium has 2 positive charges, so it feels even more positive because it gives up 2 of its ions. And it does this for stability, and it's all related to the periodic table, and if I had one I'd show you and you'd hate me for it, but you're lucky I don't. So all of these are cations because they have positive charges. How I remembered that is that the T in cations kind of looks like a plus sign for positive charge, so cations have positive charges. I'm also kind of a cat person, which is a positive thing. If you're a dog person, then it's a negative thing, so don't think about it that way. Think cat has a plus in it, and a cation has a plus charge on it. The opposite of this then, what are the ions that have negative charges? The one that tends to, or the one that travels with sodium is chloride ion. So outside in our extracellular fluid, you'll find chloride ion is our anion that we'll see out there. And we're going to talk about the movement of these 3 ions today. The only ones that are involved in an action potential are sodium and potassium. In these graded potentials that happen through chemically gated channels, chloride ion can move, potassium ion can move, potassium and sodium can move. We'll talk more about that in a little bit. The voltage-gated calcium channel is going to come in at the end when we're converting that action potential back into a neurotransmitter signal. So, okay. Those are chemically gated channels. What about our voltage-gated channels? How do they work? Because the voltage-gated channels are the ones that are going to be conducting our action potential. So we've got 2 types of voltage-gated channels. So I actually want to draw them out for you because I think that's where our lecture is starting today. So the voltage-gated channel that's going to get the action potential started is the voltage-gated sodium channel. Which I'm going to from now on abbreviate as the VGNC. And it's going to, at rest, be closed. So here we are, we're closed. But the thing about the voltage-gated sodium channel is that not only can it be closed, it's also got this little like inactivation gate hanging out here, which can like close that and lock it up for the winter so that nobody's getting in. And that's going to be really important. So, okay, so you're just sitting here at rest at negative 70 millivolts, and that is closed. Okay. What is going to open this? What's the key to open a voltage-gated channel? It's some millivoltage. So this channel is going to stay closed until there's a current that comes into this cell and brings in a positive flow so that we have cations flowing in and making our membrane potential go up to, will open at negative 55. They call it the threshold potential. The threshold potential is negative 55 millivolts. So it's 15 millivolts more positive than resting membrane potential. That's the key to open our voltage gates, is our threshold potential. So if we're thinking about the key for voltage gates, it's the threshold potential, which is negative 55 millivolts. Okay, so we'll have some graded potential in the receptive segment of the neuron. And if we have a net influx of sodium ions, and it's enough of a positive current into the cell to get us to negative 55 millivolts at the axon hillock, that is the first place that has voltage-gated sodium channels. So they're going to go from closed to open when we reach negative 55 millivolts. And what happens now through an open voltage-gated sodium channel is that sodium is going to rush in all the way to positive 30 millivolts. I'm sorry to switch markers right now, but it's driving me crazy. So sodium rushes in to positive 30 millivolts. Okay, so an open voltage-gated sodium channel is going to stay open from negative 55 millivolts to positive 30 millivolts. And then once we hit that positive 30 millivolts, it's going to enter the third stage of how it works. It's a voltage-gated channel. This is how it operates. When we hit positive 30 millivolts, it closes, but now this inactivation gate, which has been sitting over here doing nothing, is going to swing shut and block this so that there is nothing that we can do to open this channel again. That is really important because it keeps the action potential moving in one direction away from the cell body. So for our operation of our gated channels, be aware we've got two voltage gated channels that are important in the action potential. The voltage gated sodium channel, which has these three states of being: closed, opened, and then closed and inactive. Or you could just say inactive. Okay, and it's going to stay this way from positive 30 millivolts to about resting membrane potential. So we've got to start swinging back down before we're going to return to just this closed state. Okay, and we'll talk more about that in a little bit. Right now I just wanted to freshen us up and make sure we had a full understanding of everything we talked about yesterday. The other type of voltage-gated channel that's going to be important in an action potential is the voltage-gated potassium channel, and it only has two states of being. It's still going to open at threshold potential. Your book does not say that, but if you take an MCAT and you write that, or what your book says, you're going to fail that question. So this is a flaw in entry-level A&P books. They say that the voltage-gated potassium channel opens up at positive 30 millivolts. That's not true. You can only see the effect of it at positive 30 millivolts because that's when the voltage-gated sodium channel closes. So when you can no longer see the effect of sodium rushing into the cell, you can see the effect of potassium leaving the cell. So because there's a discrepancy between reality and what your book says, how I tend to phrase it is that the voltage-gated potassium channel takes over at positive 30 millivolts, because that's what's true. So we can just say, if you just want the words to say, the voltage-gated potassium channel, like all voltage-gated channels, opens at a threshold. Its threshold is the same threshold. That's what we're going to put in our back pocket as the voltage-gated sodium channels. The problem is that the whole time the voltage-gated sodium channel is open, you can only see the effect of sodium dumping in. Okay, so our voltage-gated potassium channel, it's got two states of being. It is either open or closed. At rest, our voltage-gated potassium channel, so I'll write it one time, voltage-gated potassium is spelled with a P, but its abbreviation is K, and our potassium ion is K+. So in a voltage-gated potassium channel, it can be closed, and it's closed at rest at our negative 70 millivolts. Okay, and then at threshold, we'll say at threshold, and then I'm going to know that it's negative 55 millivolts. It's going to open. Your book's going to say it's positive 30 millivolts. I'm going to say that's when it takes over. So what happens when this one is open now is positive charge is going to be exiting the cell. I know it's really weird to think about a cell becoming negative again while we're looking at a positive ion, but if we look at the direction it's going, it makes sense. So what happens when the voltage-gated potassium channel opens is potassium is going to go down its concentration gradient from high inside the cell to low outside the cell. So potassium is going to exit from positive 30 millivolts, and it actually goes all the way back down to its equilibrium potential of negative 90 millivolts. It's going to overshoot our resting membrane potential and bring us more negative. And that's going to be good. We'll talk about why in a minute. It's going to help us code for intensity of stimulus. But then what that's going to mean is that time when we're going from negative 70 downwards, our other channel is going to reset to just closed and not be inactive. Okay? So potassium is going to exit from positive 30 to its equilibrium potential of negative 90 millivolts, and then this will shut again. Okay? So those are how gated channels work. The voltage-gated calcium channel is also going to open in response to a voltage change. It also only has two states of being, open and closed. So it seems like there are multiple types, well, we know for sure there are multiple types of voltage-gated calcium channels, but it seems like the jury is still out on exactly the millivoltage that those are all opening at. So when we get down to the transmissive segment, we'll see that a voltage-gated calcium channel is going to open. It's going to open in response to the depolarizing swing, which is what sodium's doing, and then close a little bit, a few milliseconds later. So we'll get there in a minute. But as far as operation of gated channels go, it's some specific voltage that's going to get them to open up. And then when they do, their ion is going to flow down their concentration gradient. So we're facilitating diffusion and allowing a current to flow. Okay. So we left off yesterday going over quick definitions of all of these things. And graded potentials, we said, were the potentials that are short-lived. They are going to decay over time and space. Let's just talk more about these at the end of the chapter when it goes over their summation and things. So, because we did talk about them last class. These are occurring in the receptive segments. So for now, what we could say, or something new maybe that we could say for today that's going to help us move forward, is graded potentials are occurring in the receptive segment. Which two parts of the neuron are receptive to a stimulus, a neurotransmitter stimulus? The dendrites and the soma. So the only place we see graded potentials are on the dendrites and the soma. And these are the only parts of the neuron that contain neurotransmitter or ligand-gated channels. Ligand. So the only part that have these chemically gated channels. Okay, so they're going to be able to respond to neurotransmitter and cause these graded potentials. We have two types of graded potentials. Excitatory graded potentials are going to move us closer to the threshold. And those are going to be small depolarizing events where we just have a little bit of positive charge coming in. So our excitatory graded potentials, we'll say, are small depolarizing events that move the cell closer to threshold. Okay, so if we think about the receptive segment is here and our axon is here. Right here in the axon hillock is the initial zone where we're going to trigger an action potential. So if in here I get some a net positive current that gets me from -70 to -55, then I'm going to start my action potential and it's going to go. So graded potentials are occurring here in the receptive segment, and the excitatory ones are going to move you closer to threshold. The inhibitory ones are going to be small hyperpolarizing events that move you further away from threshold. So our inhibitory are small hyperpolarizing events that move you further from threshold, or that move the cell further from threshold. And it's kind of weird to do it in this order. I'm not quite sure why your book does, but we'll just go with it. Because right now, what we're going to spend an inordinate amount of time talking about is the action potential. And then at the end, we're going to come back and talk about summation of these graded potentials that results in the action potential we just talked about. We'll go with it. We can. They wrote the book that way. Just because, never mind. Okay, anyway, action potentials are going to then be constitute the nerve impulse. They're being conducted, they're being triggered in the initial segment and being conducted down the conductive segment, and will result in neurotransmitter release at the transmissive segment. So we're trying to go from a chemical message to an electrical message to a chemical message again. So our action potential is going to be the electrical message. long distance, it's all or none. It's going to be self-propagating from the initial segment all the way to the end of the axon, and it goes without decrement, and they're super cool. So let's talk about those. All right, so we just talked about the key players in the action potential. The voltage-gated sodium channel and the voltage-gated potassium channel are both closed at rest, which is negative 70 millivolts. Okay, they are both going to open at threshold. Which is negative 55 millivolts. I'm just going to put open at threshold here for the voltage-gated potassium channel, so that, because some people really get irritated because your book says it one way, even if it's wrong. Some people like to be wrong. I can't help that. I don't like to be wrong ever. Okay, so the thing that's going to happen here, this is a fast voltage-gated sodium channel. So when this opens at threshold, sodium dumps into the cell. So through this, when it opens, sodium, it's an ion channel. So the receptor is an ion channel. So when it opens, sodium rushes in to positive 30 millivolts, and this is going to inactivate the channel. So the voltage-gated sodium channel is now inactive from positive 30 millivolts. I think it's actually back down to like between threshold and rest. So we'll say to around resting membrane potential. And then what's going to happen because of our voltage-gated potassium channel staying open longer than rest is when we hit rest, this is going to return to closed. So now we can open it again if we really, really want to. So we'll say that now it's going to, it's inactive from positive 30 millivolts to resting membrane potential, and then it will return to closed, we'll say from about resting membrane potential to through hyperpolarization. And we'll talk about hyperpolarization in a minute. Okay, that's just putting into words all those silly pictures I drew up just a moment ago. Okay, so the voltage-gated potassium channel is closed at rest. It opens at threshold. At positive 30, it takes over the action potential because this closes. So we'll say when the voltage-gated sodium channel closes, this is when your book says this channel opens. It's been open all along. We just couldn't see the effect because this was open. So when the voltage-gated sodium channel closes, the voltage-gated potassium channel takes over, and potassium exits, first repolarizing the cell from positive 30 back to negative 70 millivolts, but then it stays open a little longer, so it hyperpolarizes the cell. So we'll say potassium exits, we'll say repolarizing, and then hyperpolarizing. To negative 90 millivolts. Then it closes. And it doesn't have an activation state, so it just closes. Then our leak channels and the sodium potassium pump take back over, and we return to rest, where both of our channels now are just closed. Okay? So that's what they're doing. So what are the steps in an action potential, and how is this going to look electrically? So I've written the words, I drew the state of the channels, and I've talked about the ions moving through them. So now you just have to imagine like millions of little tiny sodium ions with positive charges flooding into the cell. Okay, so we know that that's what's going on because it's an ion channel. When it opens, that's what happens. Sodium floods in. This is a thing that we can measure. You can taste sodium chloride as salt. You know this stuff, people. So it's going to sound and seem really complicated, but it's not. These are just proteins. This has no choice but to open at negative 55 millivolts. It has no choice but to close at, and when that happens, sodium has no choice but to go from high concentration to low concentration. That's chemistry. It's diffusion. So it's going to rush into the cell. This channel has no choice but to close at positive 30 millivolts. This channel has no choice but to be open, and it has no choice but to close at negative 90. So just be aware of that. There's like, I know that it seems confusing and like it's a lot, and it is, but it's really not as bad as you think. If you think, just try and break it down to the most simple level and know that there's not thoughts or thinking about it going on here. It's just the movement of ions, okay? And in the first step, we're at rest. So at rest, all voltage gated channels are closed. Let me see where our 4 is going to go to. Okay, so I actually want to draw a figure of this. This is like horribly out of proportion, but let's just imagine this is positive 30 millivolts, which is the highest we're ever going to get. We'll imagine this is 0. We'll imagine this is negative 90 millivolts, which is the lowest we're ever going to get. At rest, we're sitting at negative 70 millivolts, and we're looking for a threshold of negative 55 millivolts. Okay? All right, so at rest, stage 1, all voltage gated channels are closed. So you're at rest. You're at 70 millivolts. We see nothing interesting. So stage 1, at rest, all voltage gated channels are closed. All voltage gated sodium channels are closed. All voltage gated potassium channels are closed. I'm going to switch colors and use light blue for what happens in step 2. What's going to happen here actually is we're going to be summing up graded potentials. We'll talk about this in a little bit, so I'm going to put this red because we're talking about it. We're going to sum up graded potentials, and as long as we get enough to get to threshold, then we'll enter stage 2 of the action potential. At threshold, our voltage gated sodium channels open. At threshold, which is negative 55 millivolts, our voltage gated sodium channels open. So now sodium is going to quickly rush into the cell to positive 30 millivolts. And then this channel is going to close and inactivate. The analogy I like to use is like if the key to your door is just your key, then you stick in your key and you can open it. Okay, that's what the closed voltage gated sodium channel was like down here at rest, and it's what the voltage gated potassium channel is like. It's just closed. So all I need is the key of the threshold to open that door, okay? And then I'm open, and ions can flow. If I open my door, students could flow in as if I have that key, okay? When I get to this positive 30 millivolts and enter that inactivation state, no matter how much I hit this channel with the key of negative 55, it is not going to open. This inactivation state is like putting boards up over the door and hammering them like, with nails, so that not only is my door locked, but even if you have the key and try and unlock it, you're not getting through. I just boarded up for the winter. That's what the inactivation gate is like, and that's what's going to happen at positive 30 millivolts. So in our second step of our action potential, we call this the depolarizing swing. What's happening is sodium is rushing in from negative 55 and depolarizing our cell all the way to positive 30 millivolts. So we could write that down, that this is our depolarizing swing. And this is due to the voltage gated sodium channel. Okay? Now, here in our third step, this voltage gated sodium channel inactivates. So now the voltage gated potassium channel can take over, and we'll see the effect of potassium exiting the cell. Positive charge leaving the cell is going to make us go negative again, which is good because we need to be able to come back to rest so we can get the next signal going. So what we could say here is at positive 30 millivolts, 2 things happen. The voltage gated sodium channel inactivates, and the voltage gated potassium channel takes over. And potassium repolarizes the cell. So now when I'm repolarizing the cell, positive charge is exiting, I'm becoming negative again. When I get somewhere here between resting threshold and resting membrane potential, this channel is going to convert back to just closed. So now my inactivation gate swings over, and sure, I'm closed, but I could open it again. So that's what's important about this, like when we come back to this repolarizing swing, because now in our next step, some of these cells or some of those channels are going to take back over and hyperpolarize the cell. But if we really want to, I could open this channel again and start another action potential. That's going to be important because that's the only way we can code for stimulus intensity in the nervous system, because all action potentials look the same. So then what we'll say is we're going to repolarize the cell, and what I'll say right here is that the voltage-gated sodium channel returns to just closed. And then for our last little swing after rest, we're going to hyperpolarize, because some of those voltage-gated potassium channels are going to stay open a little longer and bring us down to potassium's equilibrium potential of negative 90 millivolts. So here we're hyperpolarizing. So we hyperpolarize. I forgot to write what's happening here is that we're repolarizing. I mean, I said it, but I didn't write it. So in light blue, this is sodium coming into the cell, depolarizing the cell. In dark blue, this is potassium exiting the cell, repolarizing the cell. And then we get back to our resting membrane potential, and so reset our voltage-gated sodium channel, and then some of those voltage-gated potassium channels are going to stay open to a little longer to draw the potential down to the equilibrium potential for potassium of negative 90. That hyperpolarizes the cell. And what that's going to do is allow for us to cause what's called a refractory period. So for our fourth stage here, we'll say some voltage-gated potassium channels stay open past rest. And hyperpolarize the cell. Okay, if you do not get another stimulus to start a new action potential, then your leak channels and your voltage-gated sodium, if you do not get another stimulus in this phase to open this again and start another action potential, then you're just going to come back to rest because of the leak channels in the sodium-potassium pump. So they always just bring us back to rest. And so then I'd be sitting here at rest again, unless I got another stimulus. And so then what's going to happen is I'll start summing up my graded potentials in the receptive segment, and if I hit threshold, I'll fire another action potential that looks exactly the same. Voltage-gated sodium channel opens up at negative 55 and stays open until positive 30. Voltage-gated potassium channel takes back over and repolarizes the cell. Voltage-gated potassium channel stays open a little longer than rest and hyperpolarizes the cell. Action potentials look the same way every time. So that's that. There are these movies that come with your book, A&P flicks. You should be able to find them through Mastering A&P, and you can look them up. Look up the one for setting resting membrane potential. It's the first one, like A&P flicks resting membrane potential, and that will show you how we get here. And then this is generation of an action potential, so look that up too. If you don't want to if you don't want to go like scraping through Mastering A&P, I probably shouldn't say it out loud, but you can Google this stuff. Not Google it, YouTube it. Look for it. If you're using a search engine, use DuckDuckGo. They don't track you like Google does, please. But anyway, get on YouTube and you could look at like A&P flicks and watch this movie. That's what we would be doing in class right now. So if you need to take a break and go grab a drink and watch this little movie before you do more physiology, do it. Okay. So propagation of action potentials. To propagate means to keep it going. And yesterday we said action potentials are self-propagating. Another thing I want to say about an action potential is, sure, we just talked about how it's got these 4 main steps: depolarization, repolarization, hyperpolarization. But a definition of an action potential, other than calling it like a nerve impulse or something like that, is an action potential is a wave of depolarization followed by repolarization. And why did I tell you that? Well, because it's this depolarizing swing that is going to open the voltage-gated calcium channel at the end. And it's the depolarizing swing that's going to open up the next voltage-gated channels downstream. So it's a wave of depolarization followed by repolarization. And it propagates away from the axon in one direction because of the behavior of our voltage-gated sodium channels. So now let's kind of like zoom into an axon. So our axon hillock is going to be the enlarged region that we have here, and it's bigger, so it's got more voltage-gated sodium channels, but it also just has a higher proportion of voltage-gated sodium channels. So here in blue, I'm going to have my voltage-gated sodium channels. And the thing is that they're all like down the entire length of the neuron, like at regularly spaced intervals, and they're followed right like next to them. We're going to have our voltage-gated potassium channels. These things are going all the way around the neuron, but I'm drawing you a cross section, longitudinal section. And so you're seeing them from the side. Okay, so our voltage-gated potassium channels are going to be our next little guys. And these are going to repolarize our cell. Okay. So what happens here in the receptive segment is I'm going to sum up all of my graded potentials. That's what's going on here, is I'm summing up graded potentials. And if I get enough of excitatory graded potentials, that I get an influx of positive charge that's going to get me from -70 to the threshold for my voltage-gated sodium channels, then these are going to open, and sodium's going to dump into this cell. What's the threshold for these channels? -55. So if I hit -55, then what happens at -55 is this opens and sodium dumps in. It just dumps in, and it's dumping in through all these. So sodium is dumping in. There's a lot of sodium. So now I'm dumping in all the way to +30 millivolts. What's the threshold for this voltage-gated sodium channel? -55. +30 is way higher than that. So this one's going to open, and sodium's going to dump into the cell and rush in to +30 millivolts, which is going to be way above the threshold for this voltage-gated sodium channel. So it's going to open, and sodium's going to dump in. So I think some books call this like a positive feedback loop. I guess I can see it, where more voltage-gated sodium channels cause more sodium to come into the cell, cause the opening of more voltage-gated sodium channels, cause more sodium to come in, cause the opening of more sodium channels. So more and more and more. More sodium in causes the opening of more voltage-gated sodium channels, which are going to allow more sodium in, which is going to cause the opening of more voltage-gated sodium channels. So that's how we're going to be self-propagating, because as soon as these guys open in the axon hillock, there is no choice but for all of the rest of them to sequentially open downstream. Okay, so that's what's going to happen. And this is a non-myelinated neuron. So this action potential has to regenerate at every little space along the way. It's fast, but it's not as fast as it can be. So it's like regenerating here, regenerating here, regenerating here. So we'll talk more about that in a little bit. Okay. So now that this one was at +30, it's going to inactivate, right? So right here, now these guys are inactive. Voltage-gated potassium channels are going to take back over, repolarizing and hyperpolarizing us. And then, but these ones were open, so it's moving down. Well, but then it inactivates. So what you can see is the area where the action potential is right now, downstream of it, we're receptive, because all of these voltage-gated sodium channels are just closed. What state are we upstream of where the action potential is right now? What state of existence are these voltage-gated sodium channels in? They are inactive. So because they were at positive 30, so they've closed and have activated, and they are waiting for repolarization and hyperpolarization to occur over here. So this part of the neuron is refractory to a new stimulus. This part is not, it's receptive. So we're going to go and we're going to propagate the entire length of the axon in one direction because we're refractory behind it as we're moving forward. Okay, so that's what I want to say about propagation of action potentials. You can watch a movie about it. What do I want to write down about it? We'll say that the action potential propagates without decrement because of the behavior of our voltage-gated sodium channels. So our action potential propagates, or self-propagates, without decrement due to the voltage-gated sodium channel. What do I mean by that? Like, one opens, sodium floods in, and opens the next. Sodium floods in, and opens the next. All the way to the transmissive segment. All the way to the transmissive segment without decrement. So it's not going to get weaker the further you get away from the cell body, because our voltage-gated sodium channel has no choice but to be open from negative 55 to positive 30, okay? So all the way to the transmissive segment without decrement. Okay, don't touch the microphone. All right. Do I want to say anything else about this right now? Yeah, watch the movie. This is what we would be doing in class. We'd be watching another movie. So you go watch the movie. I'm going to give this guy a snack, and we will finish up lecture. Okay, so all action potentials are alike. They are the same magnitude, and they, just don't change regardless of its stimulus intensity. So how does the central nervous system tell the difference between a strong stimulus and a weak stimulus? So the CNS tells the difference between a strong stimulus and a weak stimulus by the frequency of action potentials. So we can't change how strong the action potential is, we can just change how quickly it comes. So that's what we could say, is that the CNS tells the difference between a strong and a weak stimulus by increasing the frequency of action potentials. So how can this happen? Well, it happens because of those refractory periods. What refractory periods, you say? It happens because of these refractory periods we're about to talk about, the absolute and the relative refractory periods. So yeah, we code for stimulus intensity by just increasing how quick we're firing these guys. So how does that happen, and what does it look like? There's a figure in your book that shows action potentials firing on top and the stimulus intensity on bottom. So I'm just going to try and bring that to life right here. So here's like time. So like we'll see that our first chart is stimulus intensity, or our level of stimulus. And the next is the number of action potentials delivered, or action potentials being generated. So let's say you're like, here's threshold, right? Negative 55 millivolts threshold. So if you have some stimulus that's sub-threshold, or that's a low level of stimulus, then it's not going to result in any action potentials. Okay? So as the stimulus, now let's say we've got a weak stimulus. that just hits threshold. Then what we'll see is we'll start seeing the firing of action potentials that look all the same. We go from negative 55 to positive 30, then we repolarize, hyperpolarize for a minute, back to rest. Then we go from 70 to negative 55 to positive 30, then we repolarize, hyperpolarize, back to rest. Then we sum up. So if it's just threshold level, that's what would happen. We would see the set rate of action potentials. So this a weak stimulus would do something like that. So like you just faintly catch the smell of chocolate chip cookie going by or something. Then you'll see this kind of rate. Well, now what if I have a really strong stimulus? I'm in the kitchen where the cookies are baking. Now it's not going to take too long before I adapt and I'm not going to respond, but at first, or if I just walk into the house where the chocolate chip cookies were baking, then that's a strong chocolate chip cookie stimulus. So I was sitting at rest and then I walk into the house. So now what's going to happen is I'm going to start firing action potentials, and they're going to look the same. They're just going to happen more quickly. Sorry that's, you know. And why are they happening more quickly? Because this is a strong stimulus. So how is that possible that I can speed it up? Well, it's possible because of refractory periods. So refractory periods are times during which you are insensitive to a new stimulus. So during the absolute refractory period, you are absolutely insensitive to a new stimulus, and there is nothing that we can do to stimulate you again. I need this one. You can have this one. So I'm going to need a couple actually.
2?
Yeah, I'll take 2. Okay, so refractory periods. We'll say this is when you are insensitive, or we'll say the neuron is insensitive to a new stimulus. Or it's insensitive to stimulation. And during the absolute refractory period, there is no strength of stimulus that's going to get me to fire my action potentials any quicker than they're going. So there is no way during this absolute refractory period time, there is no amount of stimulation I can give to this neuron to get it to generate a new action potential. So I am absolutely insensitive to a new stimulus. So there is no level of stimulation that will create or cause or initiate a new action potential. Why might this be? Could you think of a physiological reason a neuron might be absolutely refractory? Maybe because the voltage-gated sodium channel is inactive. So all during the time that the voltage-gated sodium channel is inactive, there is nothing that we can do to start a new action potential, because it's the opening of the voltage-gated sodium channel that gets the action potential going. So that's going to be our absolute refractory period. Then we have another type of refractory period, a relative refractory period. And I'm going to write this one in a different color. So our relative refractory period, during this time, you can start another action potential, but you have to have a stronger stimulus. You have to want it more. So you walk into the kitchen and smell those cookies as opposed to just a faint whiff like going by. What's going to increase the frequency of that is that you are stimulating so strongly that you're hitting it during this relative refractory period. Okay, and what's happening here, we could say, is that during this time you can generate an action potential, but you have to have a stronger stimulus to do it. So you're relatively insensitive to a new stimulus. Thank you, sweet boy. Thank you.
Thank you for drawing right in here.
For giving me this other marker. But it has to be very strong. So you're relatively insensitive to a new stimulus, but a strong stimulus or strong stimuli can start a new action potential. So you would start increasing the frequency of firing. And what's happening during this time is this, if we look, let's look back at our chart. Let's just say, here's our action potential. We're sitting here at -70 at rest. We fire our action potential, and this depolarizing swing, when we open this voltage-gated sodium channel, and sodium's coming in, we can't open that anymore. So it's refractory to opening again. Hold on, buddy. And the whole time that it's closed and inactive, it's also absolutely refractory. So all during this phase, before we reset, we're in our absolute refractory period. I cannot stimulate this neuron again. But remember, when I get back down to around between threshold and rest, this voltage-gated sodium channels are going to return to just closed. So now, while I'm in this hyperpolarizing swing, if I really want to, I could generate a new action potential. So, because this is just closed. So yes, I'm becoming more negative. I'm still relatively insensitive, but a strong stimulus could cause a new action potential. And this is all my relative refractory period. Okay? So, if you want to think about it in numbers, when you're just like at rest and like with a low level of stimulation and we're just doing our set rate of our action potentials firing, you could think of that to go from rest to threshold, we only have to overcome 15 millivolts, or be at about negative, we've got to go from -70 to -55 and overcome 15 millivolts difference. Well, now, if I hit the neuron here and I want to open this channel, I've got to go from -90 to -55. So now I've got to overcome 35 millivolts instead of overcoming -15 millivolts. I can do it, I just have to want to do it real bad. So that is how the nervous system is going to code for stimulus intensity. So conduction velocity, how fast do we conduct action potentials? I'm not going to test you on all of the things that are going to speed it up, but what we need to talk about are the differences in continuous conduction and saltatory conduction, because this is why myelin matters. So action potentials occur only in axons, not other cell areas. So we don't have action potentials in the dendrites or the soma, and we don't have action potentials in the axon terminals, only in the axons. Action potential conduction velocities in axons vary pretty widely. The rate of the propagation depends on two things: how big around the axon is. So the larger the diameter you are, the less resistance to current flow, so you have faster impulse conductions. And then this is what I care about, the degree of myelination. So two types of conduction can happen depending on the presence of myelin. If you're not myelinated, you're going to undergo what's called continuous conduction. If you're myelinated, we're going to have saltatory conduction, which is super fast. So in continuous conduction, you have to regenerate, this is kind of what I drew a moment ago, you have to regenerate the action potential at every segment of the neuron, because you just have to. So you've got to have voltage-gated sodium and voltage-gated potassium channels down the entire length of your neuron, and you've got to regenerate the action potential here by opening your voltage-gated sodium channels and depolarizing. That'll open these ones, so we regenerate the action potential here. Voltage-gated sodium channels open, positive, stay open till positive 30. Open the voltage-gated sodium channel here at -55. It's threshold, but positive 30 is above that, so we'll open. Sodium's going to rush into positive 30. So we've got to regenerate that over and over and over and over and over and over and over again. So that's continuous conduction. When you continuously regenerate the action potential along the entire length of the axon. Continuously regenerate the action potential along the entire length of the axon. The other type of conduction we have is what's called saltatory conduction. And what happens in saltatory conduction is the action potential is propagated down the axon in a leaping fashion, where it jumps from node of Ranvier to node of Ranvier to node of Ranvier, or neurofibril node to neurofibril node to neurofibril node. So let's imagine for a moment, here's an axon, but now, and here are all my voltage-gated channels. But now I'm going to stick a myelin sheath around it. What cells myelinate in the central nervous system? Oligodendrocytes. What cells myelinate in the peripheral nervous system? Schwann cells, neurolemmacytes. If you haven't started memorizing this stuff, you're not going to be ready for the test next week. Memorize now. You need to know this stuff. So, okay, so I've got some myelin sheath here. Oop, I've got some myelin sheath here. So now what happens is I've got voltage-gated channels here. I've got voltage-gated channels here. They're mostly absent in here. I've got voltage-gated channels here, and we could imagine this is longer and it goes, further down the board. So now what happens if I reach my threshold here and I sum up to negative 55, then sodium's going to dump in here to positive 30 millivolts. And now when I'm coming through here, because I'm insulated by that myelin, I'm not really losing a whole bunch of charge. I'm going to maintain most of that. There's a little bit of decrement, but I'm going to maintain most of that so that I can, when I get here, now I'm at a voltage-gated sodium channel here, and its threshold is a negative 55 millivolts. I'm not far away from positive 30 millivolts. I've got a lot of sodium in here. So this is going to open, and more sodium is going to flood the cell, and then it's going to be protected in here. So what happens here is I have my action potential, like it seems to leap. It regenerates here and here and here and here and here at every neurofibril node, because you're insulating and protecting from the loss of charge here. So I don't have to have voltage-gated sodium channels here that depolarize the cell and open, because I'm protecting and insulating this. It's just like the black rubber or whatever it is on the outside of copper wires, protecting those copper wires and insulating them from the loss of charge. Myelin is insulating your neuron and protecting it from the loss of charge so that you can have quick saltatory conduction, where we're conducting our action potential in this leaping fashion so it's super quick. So for saltatory conduction, we'll say this is, it's very fast. And what happens is it seems like the action potential leaps, I'm going to put that in quotes because it's not really leaping, from neurofibril node to neurofibril node to node. And so it's very, very quick. I mean, just think right now, think about wiggling your big toe. You have just conducted an action potential so quickly that like you think about it and it's done. The nerve, the command is originating in your brain. And it's going down in upper motor neuron to interneuron to our lower motor neuron that's going out to our muscles in our foot. That's huge. And it happens as quick as you can think. And that's because we're going saltatory conduction of that action potential from your central nervous system to the effector. Okay, so the synapse. This is how we're going to get to, whoa. All right, so the site of information transfer in the nervous system is this spot called the synapse. And in the synapse, we've got multiple neurons interacting with each other. The first is the presynaptic neuron before the synapse, and this is going to be the one that's sending the neurotransmitter message. So we'll say the presynaptic neuron is going to be releasing neurotransmitter into the synapse. Okay, so neurotransmitter comes out of those axon boutons, or axon terminals, or synaptic knobs, or whatever it is you want to call them. So we've got these vesicles of stored neurotransmitter hanging out down here, and these are our presynaptic neurons. Okay, so cool. The postsynaptic postsynaptic neuron is on the other side of the synapse. Is it really on the other side of the synapse, or is it part of the synapse? It's part of the synapse, but these are all neurons we're talking about. So our postsynaptic neuron, here's its soma, or its cell body, and here are its dendrites. So this is the receptive segment. And it's this postsynaptic neuron that we are going to look at to determine whether or not we are going to fire an action potential. So this postsynaptic neuron is going to sum up everything that's going on from all of the presynaptic neurons talking to it and decide whether or not it's going to fire an action potential. So that's where we're going, and we're going to describe all of those potentials and stuff in a moment. But the synapse is this area where we have the axon terminal of our postsynaptic neuron and this little space called a synaptic cleft, and then either the dendrites or the soma of the postsynaptic neuron. So a synapse has three parts. One is the axon terminal of the presynaptic neuron. Two is the space right here, the synaptic cleft. And three is either the dendrites or the soma, some part of the receptive segment of the postsynaptic neuron. And I'm just going to do an arrow here because this guy's our postsynaptic neuron, and we already said that. So the synapse has a presynaptic neuron and a postsynaptic neuron. And what we're trying to get to is how we generate this action potential that's going to be conducted down our postsynaptic neuron. And the thing is that this guy's got neurotransmitters down here being stored, and they're going to be released. So this will become the presynaptic neuron for the next postsynaptic neuron. Synapses are all over the place, all over the place. This is how neurons talk to each other. So a sensory neuron in a sensory nerve talks to an inner neuron in a multipolar interneuron in your spinal cord, shoots that message up to your brain, and then we shoot it back down through a motor neuron so we can do something. Everywhere that we're transmitting a message from a neuron to a neuron to a neuron, all of that transmission is happening at the synapse. So the synapse is the site of information transfer. And it's got two neurons involved and then the space in between them. So let's talk about different types of synapses we can have. So what we're really going to spend a lot of time talking about right now is chemical synapses. These are the most common synapses, so we're going to go over them in a lot of nitty-gritty detail. There are also electrical synapses, but they're pretty rare. So we will briefly address them. Here's a brain break for you. This is the first season I've had seasonal allergies, and we have coronavirus running rampant. So let me tell you, when you're sneezing and coughing in this environment, people look at you like you have the plague. So, all right. Anyway, I digress. Chemical synapses are the most abundant type of synapses in the body, so we're going to spend a lot of time talking about them. Synapses are the site of information transfer. And so why this is called a chemical synapse is because we're using a chemical, a neurotransmitter, to transmit information from one neuron to the next across the synapse. So how this is going to happen is an action potential is going to come down our presynaptic neuron. Okay? So an action potential travels down the axon of the presynaptic neuron. We just talked about action potentials, so you should have a little bit of an idea about what that means. It means that we had this wave of voltage-gated sodium channels opening and allowing this really massive influx of sodium into the cell, followed by this wave of voltage-gated potassium channels opening, allowing for an efflux of potassium out of the cell, causing this depolarization and this repolarization and this hyperpolarization down the cell. So that action potential is traveling down the axon of the presynaptic neuron. When it gets to the axon terminal, that's this enlarged region at the end. There are voltage-gated calcium channels here. I have mentioned them, but we have not explicitly talked about what they do. Now, when our action potential is coming down here, the depolarizing swing of that action potential is going to cause those voltage-gated calcium channels to open. So let's write that down and see if we can wrap our head around what that means. So the depolarization opens voltage-gated calcium channels in the synaptic knob, or axon terminal, or whatever it is you want to call it. What's going to happen now is we've talked about vesicular transport a couple times and said that exocytosis is this process by which you take a membrane-bound vesicle, fuse it with the plasma membrane, and then dump the contents to the extracellular space. That's how neurotransmitters get released. So if you were to zoom into the synapse, you have all of these vesicles that are made of the same stuff as the plasma membrane. So why is it that we're not just continuously dumping neurotransmitter into the synapse? The reason is there are regulatory proteins here holding those synaptic vesicles in place. Now when this action potential gets down here and opens these voltage-gated calcium channels, calcium's going to come in here. Calcium is a bigwig. Look at this. It's got 2 positive charges. That's a big deal. So anytime calcium comes into a cell, stuff is going to change. Stuff is going to happen. It's going to move regulatory proteins and really amazing things happen. In the nervous system, the really amazing thing is we're going to have vesicular transport, which isn't super amazing. It's exocytosis. But we release neurotransmitter now to this synapse, and this electrical message that was an action potential gets converted to a chemical message that's a neurotransmitter. So the neurotransmitter is going to be released into the synaptic cleft, and that will start our next kind of part of the of what's going on at the chemical synapse. So our action potential is going to travel down this axon in the presynaptic neuron. The depolarizing swing is going to open these voltage-gated calcium channels in the synaptic knob. Calcium is going to bind with all these proteins and allow for the exocytosis of neurotransmitter to the synaptic cleft. So calcium binds, or we'll say that calcium enters the neuron and neurotransmitter is released by exocytosis. So now this neurotransmitter is going to enter the synaptic cleft, cross the synaptic cleft, and bind to its specific receptor on some part of the receptive segment of the postsynaptic neuron. And it's got to be the specific receptor for this neurotransmitter. So for stage 3, what we could say is happening here is that neurotransmitter enters synaptic cleft, it's going to bind with receptors on the postsynaptic neuron, which is this guy right here. And now, so our neurotransmitter has entered the synaptic cleft, it crosses, it binds its receptor, and now that receptor is a ligand-gated ion channel. So when the neurotransmitter binds, this protein is going to change shape and open up, and ions are going to flow down their concentration gradient through it. And that could be into or out of the postsynaptic neuron. So our 5th step, we could say, is that this binding with its receptor results in the opening of an ion channel. So we'll say an ion channel opens and a graded potential develops. Okay, so now what's going to happen is the neurotransmitter is going to be degraded by an enzyme in the synapse or reuptaken, and parts of that will be reuptaken, or diffuse away in the synaptic cleft, and transmission will stop. Okay, so that's kind of our last, or like the final step. We could say transmission at the synapse ends when neurotransmitter is broken down, reuptaken, and or diffuses away from the synapse. So that is how information is transferred at a chemical synapse. We have an action potential coming down our presynaptic neuron, resulting in the release of a chemical, the neurotransmitter, that's going to cross the synaptic cleft, bind with its receptor on the postsynaptic neuron, and open an ion channel. The opening of these channels is going to be what sums up to determine whether or not that postsynaptic neuron conducts an action potential. So that's where we're getting. Okay, so that's how chemical synapses work. Electrical synapses are when our presynaptic and postsynaptic neurons are joined by gap junctions. And so if you recall from day 1, I think, in class, we talked about cell junctions, and you could see that in a gap junction, there was like almost instantaneous passage of chemicals between those pores from one cell to the next. So that's what's happening in electrical. So that's what's happening in an electrical synapse. And those are very rare in the body. You find them in places where you need to do really rapid movement. So like in some of the neurons that control the movement of your eyes, you find electrical synapses. So we'll just say they're rare. Our neurons are joined by gap junctions, and they are very fast. Okay. All right, so why do we talk about all this? Well, okay, we've talked about synapses. The chemical synapse is what we really care about. And in our chemical synapse, our presynaptic neuron is going to be releasing some neurotransmitter that's going to bind some receptor and either move us closer to or further away from potential. So threshold potential. So we'll either get closer to an action potential or further away in that receptive segment. The job of the axon hillock is to add it all up. So why don't we just draw the whole neuron up here so that we're all on the same page. What we're talking about is a chemical synapse now, and what's going on here at the synapse, at this receptive segment. What's happening here to determine whether or not our postsynaptic neuron is going to fire an action potential. So the question is, are we going to fire an action potential in the postsynaptic neuron? And that will be answered by whether or not we are overcoming that millivolt requirement to hit negative 55 millivolts right here. So we can have excitatory synapses where our presynaptic neuron is releasing a neurotransmitter that binds to a receptor on a postsynaptic neuron and opens an ion channel that moves us closer to threshold. That's excitatory. So excitatory synapses cause what are called excitatory postsynaptic potentials. We're looking at the potential that's being formed here in our postsynaptic neuron. So this is our postsynaptic neuron, And what we're measuring is the potential that's happening here. So an excitatory postsynaptic potential moves you closer to threshold. So EPSPs are excitatory postsynaptic potentials, which I'm from now on going to abbreviate as EPSPs, and these move the neuron closer to threshold, or towards threshold. What's threshold? Negative 55 millivolts. Okay, so our postsynaptic neuron is sitting here at resting membrane potential. What's that? Negative 70 millivolts. Okay, so we're sitting at negative 70 millivolts and we're trying to get to negative 55 millivolts. Okay, excitatory postsynaptic potentials get you there. And how we do it is we open an ion channel that's going to allow for some kind of positive charge to come into this cell. So the neurotransmitter binding is going to open a chemically gated channel. Specifically, this is a cation channel. This is a very specific channel. It's not a sodium channel, because sodium channels only let sodium through. It's not a potassium channel, because potassium channels only let potassium through. This is a cation channel, and it's going to allow potassium to exit the cell, but it's going to allow sodium to enter the cell, and sodium is kind of always like more. So more sodium is going to come in than potassium leaves, so the net influx of sodium is going to move us closer to threshold. So we'll say we have some potassium out, but we have more sodium in, and it's the net influx of sodium that is going to move us closer to threshold. So a potential or an event that moves us closer to threshold, is that depolarizing or hyperpolarizing? This is a graded potential. This is going to be a small, graded, depolarizing event. So as long as neurotransmitter is bound to this type of channel, and it's open, we'll have a little bit of potassium out, but we'll have a lot of sodium in, and we'll be bringing positive charge into the cell. But as soon as the neurotransmitter is degraded, or as soon as the signaling here stops and the neurotransmitter is degraded or taken away, this signaling stops, and our leak channels in the pump take back over, and we return to rest. It's graded. It's going to decay over time and space. So just one excitatory postsynaptic potential isn't going to be enough to get you there. We'll talk more about summation in a minute. So what an excitatory synapse is the synapse that occurs when a neurotransmitter binding opens a cation channel. And that's going to result in an excitatory postsynaptic potential, because we'll have this increase in this influx of positive charge as sodium, we have a net influx of sodium into the cell. This is going to, create this excitatory postsynaptic potential and move us closer to threshold. Okay. Well, it's also possible that we move further away from threshold. So I'm actually going to just leave this blue stuff over here for excitatory, because we want to also think about inhibitory and what's going on here. And I'll tell you what it says under here. You can read it in your book, you can pause, whatever. Inhibitory synapses and inhibitory postsynaptic potentials occur when the neurotransmitter binding to the receptor is going to open a chemically gated channel, but now we're going to have an event that moves us further from threshold. What type of event is that? Hyperpolarizing. So for our IPSPs, or our inhibitory postsynaptic potentials, what's happening is we are moving away from threshold. So our neurotransmitter binding to a receptor is going to open chemically gated ion channels that move the postsynaptic neuron away from threshold. So these are graded hyperpolarizing hyperpolarizing events. And they're inhibitory because they move you away from threshold. So we actually have two types of channels that can be opened to cause an inhibitory postsynaptic potential. We can open a potassium ion channel, and we can open a chloride ion channel. Both of these events, if the neurotransmitter's receptor is an inhibitory synapse here, if the neurotransmitter here binds to a chloride ion or a potassium ion channel, those channels are going to open and move us further from threshold. We're trying to get to -55. So what's going to happen is we're going to become more negative. We'll have these graded hyperpolarizing events that make us more negative. So why would these types of channels do that? Why would they make us more negative? What way does potassium flow through a channel? It's a positively charged ion. So when we open a chemically gated, ligand-gated, neurotransmitter-gated potassium channel, what way does potassium move? It moves out, right? So we've got positive charge exiting, and it moves out because potassium is higher inside the cell than out. Where's chloride ion higher? I briefly mentioned it a couple times. Chloride ion is found with sodium, and sodium is higher outside the cell. So chloride ion is also higher out as sodium is higher out. So if we open a chloride ion channel here, what's going to happen? Chloride ion is going to move down its concentration gradient from high outside to low inside, and a negative charge coming in is going to be another hyperpolarizing event that moves us away from threshold. So either way, opening both of these channels is going to result in this inhibitory postsynaptic potential, and it's inhibitory because we're getting further away from threshold. The threshold we're trying to reach is -55 millivolts right here at the axon hillock. Okay? So this is something that's important to be aware of. I could dump neurotransmitter on the axon all day long, and I will not get anything excitatory or inhibitory, regardless of it, whatever. Why? Because there is not a neurotransmitter-gated receptor here. So I could dump chemical on this part of the neuron all day long, and nothing's going to happen because there are no chemical receptors. What kind of receptors are here? Voltage-gated sodium and voltage-gated potassium. Same thing here. I could dump neurotransmitter all day long and nothing is going to happen because what type of channel is here? Voltage-gated calcium. Now when the calcium is in here, neurotransmitter is going to be released, and that's what's going on here. Neurotransmitter is released, and then it's going to bind to ligand-gated receptors here. This is the only place I find ligand-gated receptors, which are ligands are chemicals, or ligands are chemicals, chemicals are neurotransmitters, and we find them in the receptive segment. So excitatory and inhibitory postsynaptic potentials occur in the receptive segment of a neuron, and those things are going to sum to determine whether or not this postsynaptic neuron fires an action potential of its own. So how do we sum? Summation is adding up. We can sum up in time, or we can sum up in space. What does that mean? Let's leave this neuron up. Okay, so just for a minute, we're going to think about, let's just imagine that our light blue synapses here are inhibitory synapses, and our dark blue synapses here are excitatory synapses, or that's what we got going on. We can have an excitatory synapse right here, right on the soma directly. So, and the reason it is excitatory is because the specific neurotransmitter receptor right here is a cation channel. And the specific neurotransmitter receptor right here is a cation channel, okay? And then I can also have some presynaptic neurons that are going to create an inhibitory synapse. And this synapse is inhibitory because the receptor right here is going to open a chloride ion channel. And this one is inhibitory because the receptor right here is going to open a potassium ion channel. Okay? So what I'm summing up are postsynaptic potentials. And I can sum those up in time, or I can sum those up in space. And first, let's think about what happens so that we can get to threshold and get an action potential. So, okay, how are we going to sum up in time? The only way we're going to get an excitatory postsynaptic potential that's going to move us closer to threshold is by opening this cation channel. So if I just have one excitatory postsynaptic potential, sure, I'll get this net influx of sodium, but it's not going to be enough if it's just one impulse. It wouldn't be enough to overcome this 15 millivolts that's required all the way down here. Why? Because only a little bit of sodium is going to come through here. If it's just one neurotransmitter binding, I'm going to have a net influx of sodium. Remember, through the cation channel, potassium is also leaving. But I get this net influx of sodium. But if there's not another impulse here, then the sodium's going to be pumped back out by the sodium-potassium pump, and I'll return to rest. So just one's not going to be enough. If I want to get enough excitatory postsynaptic potentials out of this synapse to overcome the 15 millivolt difference to open up this, our voltage-gated sodium channels at threshold, then what I'm going to have to do is I'm going to have to fire this one, fire this one, fire this one, if I want to do it temporally. So what I can do for temporal summation, we could say, is when we add up our postsynaptic potentials in time. And how do I do that? I increase the frequency of stimulation here. So instead of just having one impulse from here that causes one quick excitatory postsynaptic potential, now I'm going to have one, but then I'm going to quickly stimulate it. So let's say I stimulated it here and I got this impulse. Now I'm going to stimulate it here and I'm going to stimulate it here. So I go bam, bam, and I sum up. So that I had one excitatory postsynaptic potential, but before I could come back to rest, I had a second one that added on top of that, and that was enough to get me to threshold and shoot my action potential all the way up to positive 30 millivolts. And then that's going to conduct down the entire length of the axon without decrement till we get down here and we open up our voltage-gated calcium channels to start it all over again. Okay? So that's what's happening in temporal summation. You could also temporally sum some inhibitory postsynaptic potential, sure. So if I have like, if I have just one inhibitory postsynaptic potential, I'm sitting here at rest at negative 70 and I have one and I'd hyperpolarize, but then I'd come back. So here's my site of stimulus for my inhibitory postsynaptic potential. Okay, but if I increase the frequency of stimulation here and I hit it here and I hit it here, then what'll happen? I'm sitting here, I come back to rest and I get hit here and I dip. And I get hit here and I dip even lower. And I move even further away from threshold. Okay? So I could sum up both types in space. Now it's just going to be even harder to get to threshold here, even harder for this postsynaptic neuron to fire an action potential. All right, well, what's spatial summation? This is when I sum up in space. So when I have multiple presynaptic neurons stimulating one postsynaptic neuron, what's going to happen here? This postsynaptic neuron has to sum all that up over space and say, okay, I've got one inhibitory postsynaptic potential occurring here. I've got two excitatory postsynaptic potentials occurring here. Is that going to be enough to overcome it? Well, we got to figure that out. So for spatial summation, we could say this is occurring when our postsynaptic potentials add up in space, or we could say more than one presynaptic neuron is firing onto one postsynaptic neuron. Okay, so first let's think about this cation channel here again. And look, there's a neighboring cation channel here. We've got one cation channel synapsing onto the dendrite, onto a dendrite, and one cation channel synapsing onto the soma. So if we just have one of them going, it's going to be that little impulse that's not enough. But if this one fires and this one fires, then they can sum together. So if just one went on its own, let's say we had a graded potential that was like that. But since both of them are firing at the same time, then I get enough. and it's enough to hit my threshold and trigger my action potential. So if I were to sum up spatially, let's say I had one chloride ion channel open at the exact same time one cation channel opened, then they would cancel each other out and I would still not generate an action potential. If I had one chloride ion channel open, so an inhibitory synapse activated here, and one potassium channel open, and an inhibitory synapse activated here, and one excitatory postsynaptic potential because of an excitatory neurotransmitter binding here, then would I move closer to or further from threshold? That would be spatial summation of those three synapses. Do you think I would move closer to or further from? I would move further from, because like these two would cancel each other out, but this one's open as well. So positive charge is exiting and I'm moving further from threshold, okay? So that is how we sum up in time and space. And in reality, we're summing up both ways all the time, all over the nervous system. So it's an electrical storm in there and it's amazing. All right, let's see. I have a couple questions for you. And I want to say we're almost done, but we're probably not. Okay. I think I already did this one. Did I? Yeah, I did. Skipping it. An excitatory neurotransmitter will open A sodium channel, B, a cation channel, C, a potassium channel, D, all of the above. Yeah, the answer is cation channel. And now some of you are like, well, wait a minute, I thought that sodium is what caused an excitatory postsynaptic potential. It's a net influx of sodium into the cell, true, but it's a cation channel that opens. So we're having potassium go out of that channel and sodium come into that channel, and it's the net influx of sodium that causes an excitatory postsynaptic potential, okay? So, but this is asking about a neurotransmitter. An excitatory neurotransmitter opens a cation channel. The neurotransmitter in the previous question was excitatory because A, its receptor opened the cation channel, B, it had a positive effect on the cell, C, some neurotransmitters are always excitatory, or D, all of the above. The answer is A. The reason that neurotransmitter is excitatory is because we open the cation channel, and that's net influx of sodium is going to be what causes the kind of buildup in positive charge that should move us away from rest and toward threshold. Okay, we are almost done. So neurotransmitters, there are a lot of neurotransmitters. There are like 50 or more. We know probably next to nothing about the nervous system. We sure think we know a lot, but we are learning things all the time. So just be aware of that. Science changes, life changes. Welcome to it. So 50 or more neurotransmitters have been identified. Most neurons make 2 or more. Some make only 1. Neurons can have several influences. The influence that they have is determined based on what type of receptor they're interacting with and what's happening then in the postsynaptic neuron. So usually if neurons are releasing more than one neurotransmitter, they are released at different stimulation frequencies. The way we classify our neurotransmitters is based on their chemical structure. That's chemistry. Do I like chemistry? No. Am I going to test you on that? No. And their function. Their function is either they're excitatory or inhibitory. They either move you toward or away from threshold, right? That's good enough. Why do I bring it up then? Because I want you to know a little bit about neurotransmitter receptors. Why? Because we have just been talking like ad nauseum about channel-linked receptors. When a neurotransmitter binds a channel-linked receptor, a channel is going to open, and that channel opening is going to allow for ions to move down its concentration gradient. So channel-linked receptors are receptors for specific neurotransmitters that open some ion channel. What's an ion channel? It's a protein. See, we're building on everything you already know. You talked about this in Gen Bio. We went over it already in class. This is a protein that facilitates diffusion. What does that mean? It's going to allow for an ion to move through it from high concentration to low concentration. So it allows for an ion to move through it down its concentration gradient. So from high to low. An ion has a charge, so if we're moving, I am causing a current. That is so cool! I am sending electrical messages around my body. It's awesome. So, and it's just because of a protein that I stuck in the receptor of a cell. Love it. That protein is a channel-linked receptor, specific for a neurotransmitter, okay? I just totally geeked out on that. It was so exciting, and I hope you were with me, and I hope you're just like, oh, it all makes sense now. Oh, look, I could draw it. It's a light bulb moment. Oh! Okay. Man, I hope you all had that moment with me. Otherwise, I just look ridiculous. I probably look ridiculous all the time. Okay. We can also have neurotransmitters that bind another type of receptor called the G-protein coupled receptor. We'll talk more about G-protein coupled receptors next semester too when we look at hormones, but this is a different type of receptor. And what happens with a G-protein coupled receptor is that your neurotransmitter is going to bind out here on the extracellular side. Sorry, light bulb, I don't know if we're having that moment with you now. So on the extracellular side of our neuron, our neurotransmitter is going to bind, and it's going to activate its receptor that's hooked up with this thing, hold on, called a G protein. So when the neurotransmitter binds, it's going to activate the G protein, and the G protein is going to go and activate another protein. And it's like if you're G stimulatory, you're going to activate another protein called adenylate cyclase, and it's going to pump out this second messenger. The point is we're getting second messengers. But if it's adenylate cyclase, it's going to pump out, if it's the GS protein, it turns on adenylate cyclase, kicks out the second messenger, cyclic AMP. That's the most popular one in the body. So yeah, so we've got different G proteins that are going to activate different secondary proteins in the cell membrane, and those proteins are going to pump out what are called second messengers. The most popular one is this one called cyclic AMP, and what it's going to do is cause a cascade of events inside the cell. So you'll start turning on enzymes that start speeding up reactions and start doing stuff. So what's cool about G protein coupled or G protein linked receptors, usually I see it as GPCR. So GPCR, G protein coupled receptors or G protein linked receptors, is that the binding of one neurotransmitter here can cause a huge cascade of events inside the cell. So that's the second type of neurotransmitter receptor we have. That wraps up everything I have to say to you from chapter 11. So some of the material is copyrighted by Pearson, some of the material is copyrighted by me. Yeah, I look forward to seeing you in the Teacher Helping Students Succeed discussion page and in review. Talk to you soon.